Well, thank you guys for staying. I'm neither do muscular dystrophy or CRISPR-Cas9, so I appreciate you guys staying and hanging out, particularly on the last day of the conference. All right. Just a disclosure, I'm a member of the Scientific Advisory Board, and I'm a shareholder in Genescience. It's a company that's working to develop gene therapies for OA and connective tissue disorders. So just to give you a brief introduction about osteoarthritis, it's not an orphan disease by any stretch. It affects about 40 million people in the United States alone. There's no treatment for it. Existing medicines are basically to treat the pain. In OA, it's a chronic erosive condition where you lose the cartilage substance over the surfaces of the joints that cushion the knees or cushion the joints during articulation. You lose this cartilage substance and you build up subchondral bone and osteophytes. Over time, the joint is pretty much destroyed, and the only treatment is joint replacement surgery or implantation of a prosthetic joint. And I think last year, or approximately 700,000 total knee replacements occurred in the United States alone. So there's nothing available that can stop this erosive process in osteoarthritis. So we had the idea it's pretty well accepted that interleukin-1, it's a primary inflammatory mediator. It's thought to be the primary driver of pathogenesis, or at the cellular level in this disease. And so that if we were able to inhibit the effects of IL-1 signaling in the joint, we could do some good in this disease environment. We could theoretically delay the progression of disease, as well as have some reduce the pain and improve mobility. So the protein that we wanted to use we wanted to deliver a naturally occurring protein. It's interleukin-1 receptor antagonist. You got IL-1, it binds and it recognizes and binds to signaling receptors on the cells, but IL-1Ra is a competitive inhibitor of IL-1 signaling, so it binds to available IL-1 receptors and essentially blocks their signaling. The thing about this is you need to make a lot of IL-1Ra. You need to outcompete the existing IL-1, and IL-1's a pretty potent signaler. So you have to have anywhere from 100 to 1,000-fold greater concentration of IL-1Ra in the tissues than you do IL-1. So basically, it's a pretty straightforward approach that we wanted to use to try and treat this. If we could deliver the coding sequence for IL-1Ra under independent control, in this case a CMV promoter, we could deliver it to cells that line the joint, and as well as its synovial cells, as well as cells in the articular cartilage. We could convert those cells to factories for IL-1Ra production. They would just continuously produce and release IL-1Ra into the joint, and it would block IL-1 signaling, and it would be a therapy that would last for a long time. One of the problems, again, with trying to treat joint disease is it's hard to get drugs in there, and it's hard to keep them there. A systemic delivery approach, you don't achieve therapeutic doses inside the joint. If you inject it directly, it's cleared within 30 minutes to an hour. So the idea of a local gene therapy would overcome these sorts of barriers, have long-term local expression concentrated at a site of disease. So we'd done some earlier work with various vectors, landed on AAV. It seemed to work pretty well. But in some cases, when we were working in rodent models, we would look at the biodistribution, and in a lot of cases, when we would inject the vector into the joint space of the rat, we would find that there would get transduction of the adjacent muscle tissue, and it would tend to leave the joint. And we thought, this is not really what we're looking for. We'd like to have everything contained within the joint. We wanted to limit the escape of the protein from the treated joint, because you have the potential to inhibit inflammatory signaling systemically, and that was just counter to what we were looking to achieve. So we thought, well, maybe the problem is we're just looking, we're working tissues that are just not representative of something that would be like treating a human. The little tissues inside a joint of a rat, they're paper thin. It's easier for vectors to transverse the articular tissues and get out into the circulation or in the regional tissues. So we've got to move into a bigger system. We've got to move into something more relevant. We need to answer some basic questions about whether this is going to have any merit whatsoever on treating disease on a human scale. So, so we selected an equine model for several reasons. One is that they are four-limb joints there. The carpus and the metacarpal phalangeal joint are sort of the ankle. Sort of the knee and the ankle analogs of humans. Their joints, they're similar in size and volume to the human knee, a common site of OA in humans. Plus, these are joints that naturally get OA during racing and training. So they give us a source of naturally occurring disease to work with. Another good thing about it is because of the large volume is you can aspirate fluid, synovial fluid from the joint on a regular basis, and you can look for evidence of transgene expression in the synovial fluids that you recover. And also it would give us some basic information about what it's like to treat humans, how reasonable it is, what would be the safety concerns we might have if we're going to go into human trials. And something you have to think about is OA, unlike DMD or cancer or anything like this, is OA. It's osteoarthritis. You take ibuprofen. That's sort of what you do. Nobody dies from it. So it's a treatment where we're going to have to be fairly conservative with our approach and also take extra precautions and err on the side of safety. And another benefit is that because horses develop OA, the treatments that we develop in this system would be good for veterinary applications as well. So initially we had just a couple of basic questions to ask. I mean, could we, with an AAV vector, deliver enough transgene and get enough expression where we can, how much can we make? Can we generate a meaningful level of expression in a human-sized joint? And another thing is I showed you the initial slide is how the pathology of the disease changes the tissue itself. So if we deliver a virus into that disease context, how does that impact things? How does that affect transgene delivery, expression, and biodistribution of the vectors? So initially we just did some simple dosing studies inside the joints of healthy horses. We did a 10-fold escalation from 10 to the 11th up to 10 to the 13th. And we saw, a dose increase, but as we increased the dose that we added, the increase in gene expression was not proportional to the amount of virus that we were delivering. In this case, these are 10-fold increments, and at the last two, we're only gaining about a 30 to 40% increase in IL-1Ra production for a 10-fold increase in virus. So we used this 10 to the 13th dose as our working clinical dose because we figured if we went too much higher, we weren't going to get much return on our investment, expression versus dose-wise. So we did some initial studies. So let me back up. In this case, when we delivered, we could elevate the endogenous levels of IL-1Ra 50 to 100-fold, and we could keep them maintain that level of expression for over 6 months just in healthy animals. So that was very encouraging. We could, in this case, we could deliver a homologous equine transgene, and it would persist, well, at least indefinitely, at least for the duration of this protocol. When we compared, we wanted to compare gene delivery in the context of an OA joint versus a normal joint. So what's it look like? And so this is normal healthy joint. Now this is a carpal joint. It's really similar to the wrist rather than the knee, so it's got more bones in it. But basically, when you get OA in this joint, you develop the osteophytes, you lose the cartilage surface, the bones fractured. I say in the horses, it's a disease very similar to what you see in humans. And when we tracked, in this case, we just delivered an AAV vector with GFP just to see where the virus goes, what are the transduced cells locally and systemically. Inside healthy joints, we could see a reasonable amount of gene transfer in the synovium, went several layers thick in the synovial lining. But in the OA joint, we saw a tremendous increase in GFP fluorescence throughout the synovial lining. A lot of that had to do with seemed to be increased cellularity in the synovial lining, and also it appeared to be from increased activation. When we looked at the cartilage, that's where we saw the greatest difference. In healthy joints, we barely see a flicker of a green cell. However, in the OA joints, we saw lots of places where there were really hot spots for GFP expression throughout the cartilage, suggesting that, or at sites that had the most damage or that suffered the most, seemed to have by far the highest levels of GFP expression. Not necessarily due to a loss of the cartilage matrix, but it seems to be more due to changes in activation and metabolic activity of locally increased disease activity. When we looked at distribution levels, though, even though we saw much greater gene expression in the OA joints, when we looked at the distribution of viral genomes within the joint, we didn't see a significant difference between the two. But encouragingly, when we looked at tissues, extra-articular tissues, we saw a few genomes here and there, but for the most part, over 99.9% or 0.8% were contained within the injected joints. So we wanted to see, all right, so we can put things in there, we can get somewhat persistent transgene expression. Is that enough to do anything? Is it a functional level of protein expression? So we initially tried a short-term OA model, and in this case, we surgically generate a fragment, an osteochondral fragment, off the radiocarpal bone inside the carpal joint of the horse. And it's very similar to the type of injury that would occur naturally during racing and training. Initially, we wanted to do it in naturally occurring disease, but the NIH wouldn't give us the money for that. So they said it would be too variable, so we had to switch to this model where we could surgically generate an injury that would be consistent or try to be consistent among different animals, and we could, the animals, the disease progression would be of a similar timing. So in this case, so for this model, at least, we generated the osteochondral fragment. A couple of weeks later, we injected the vector into the joint with the injury, and then we trained the animals for 10 weeks on a treadmill. So like I said, this is much like a racing injury where you generate the fragment and then the animals train on it, and the training is what develops the pathologies that are associated with the OA model. And then after the end of 12 weeks, we would go in and we would arthroscopically repair the injury and return the horses to the research herd. But in the meantime, we did MRIs at the start and the end of the procedure, and we would aspirate synovial fluid on a biweekly basis. And this is just to give you sort of an idea of the scope of what it's like to work with horses as an experimental model or experimental animal, but it's quite an undertaking. So when we looked at gene expression inside the joint, what was interesting is that in the joints, relative to animals that were healthy, which we saw between 40 to 50 nanograms per mil of IL-1Ra, we saw much greater levels, 3 to 4 times higher on average in the animals with the injured joints, which was consistent with what we've seen with the GFP expression. When we deliver into a diseased joint, we got a considerably higher level of GFP expression, and it ties in here. When we looked on so this is mean levels. When we looked on a per animal basis, though, we saw a lot of variability in gene expression. But when we compared that variability to the total pathology score by MRI at the time of injection, we saw that the transgene expression, the variability in expression, directly correlated with the level of pathology at the time of injection. There was, however, an outlier. We still can't, this guy here, have no basis for why he was out there, but that's just the way it worked. But for 9 out of 10, there was a very strong correlation between disease pathology and levels of expression. We didn't see, in this case, is just IL-1Ra levels in urine and serum, and there's 2 different groups, treated and untreated, and we saw no real differences between them, so we didn't see any evidence of IL-1Ra protein leaving the joint and altering circulating levels. When we looked at lameness, which we use as an index of joint pain. Horses move, their gait is based on symmetry, and when they hurt, that symmetry changes, and you can measure that either by eye, subjectively, or by motion analysis. And we've got pretty good agreement between the two. Towards the end of the study, we started to see some statistically significant differences by both assays, by both subjective and objective evaluations of lameness. And then when we looked at MRIs, we could see some interesting differences between the 2 groups. On the top are the treated animals. These are multiple MRIs from just different views. And on the bottom are the controls, and I won't bore you with all of the details. But for the most part, what we would see is a reduction of synovitis, synovial proliferation, reduction of joint effusion. But we also saw improved repair of the actual defect itself, both at a macroscopic level as well as histologically. So that was, and yeah, that's what I was going towards here, is that in the joints that were treated, we saw better repair of the actual fracture itself, better quality bone. The cartilage itself near the site of injury showed some improvement, but it just didn't quite reach statistical significance. But overall, what we saw in this particular model was about a 25% improvement over the untreated animals, which, considering we're only blocking one cytokine, and it's a condition that's driven by a physical injury, we thought that was pretty good, because that's what you have to consider in humans. OA is a very complicated disease. You can get it from an old football injury, or you can get it from being overweight or malalignment of the joint. Those things aren't necessarily going to go away. When you go to treat this, you can only block the cellular signaling and hope for the best. So the fact that we're able to see any sort of positive effects in this type of environment it was very encouraging. So so what we did in this first study was basically sort of a prophylactic type of approach, where we would cause an injury, and then soon after the injury we deliver the vector, and then train the animals that would drive the OA pathology. And it was, again, it's a very short term, 12 weeks is the duration. But what we really were interested in is if we came in at this point, after the animals had already had some sort of established pathology or established disease, could we come in at this point and have any benefit? And would it go? How far would it go out downstream? And so we set up a different type of a protocol, where in this case, the time frame here that we used in the original study is reduced to here, where we would generate the osteochondral defect. We train the animals for 10 to 12 weeks, as we had here. In this case, it's a 5-day-a-week athletic training schedule, and it leads to the OA pathology. So in this case, at the end of the protocol here is where we wanted to treat, and then we wanted to follow the animals downstream for a period of a year. The problem is we couldn't continue to athletically train the animals 5 days a week on a high-intensity schedule because it would just be too severe, and IACUC wouldn't approve it. The veterinarians we work with wouldn't do it. So what we have to do is in order to get a chance to take a look at this long term, we had to reduce the intensity of the training to a 3-week schedule, three times a week schedule, which would maintain the level of disease, but it wouldn't progress to an inhumane type of level. but nonetheless will give us a chance to look at the long-term effects. And so in this case, when we deliver the vector post-injury, post-OA induction, and follow it out for 12 months, we found, very similar to what we'd seen in normal animals, a very similar level of gene expression, anywhere between 30 to 40 nanograms per mil, and it lasted for at least 13 months. When we look at the individual animals here, they're far less variable than what we'd seen when we delivered into that acute injury context. So it's much more predictable than what we'd seen, post-injury or post-acute injury. There's some variability here, but a lot of that has to do with just sampling our pulling synovial fluid out of horses is not that easy all the time. When we looked at the effect of treatment on lameness, we saw in this case is where we did motion analysis, and we saw a gradual improvement in joint lameness, but over a period of months. And towards the end, after about 3 or 4 months, you begin to see a decline in lameness that persists throughout the end of the protocol. Where the animals that didn't receive treatment, their lameness hovered at about the same level throughout the duration. When we looked at the MRIs, what we see is in the controls, starting level and at the end level, the level of pathology was about the same. Here's the osteochondral fragment. It's here as well. There's the level of synovial effusion. This is just a particular animal here. It's a little bit easier to see. It's why I selected it. It was quite swollen and a fair amount of edema at the time of treatment, but at the end of the 12-month protocol, it reduced considerably, and there was good repair of the fragment. If you just look at the scores overall, there were several. This is what we had to do is normalize to the level of pathology at the time of treatment, and we see in the red bars the animals that were untreated. Many of the parameters, either hover at or right around the starting level, where many of those in the treated animals drop considerably at a significant level, particularly, again, the synovial proliferation. We saw improvement in the fracture itself, and overall total pathology, about a 25 to 30% reduction in total joint pathology by MRI. When we looked at the safety and the biodistribution, we did we had 2 arms of the safety study, and these were done by an independent toxicology group at UF. So basically, after we did the injections, they took over. But we had 2 arms. The first one was a long-term study where we took the animals we'd used in the 12-month efficacy study, euthanized those, and looked at those for for the toxicology as well as the biodistribution. And then we had a acute where we treated the animals, and then 2 weeks post-treatment, they received, at treatment, there was a clinical dose and a 10x dose. The 10x dose would be the worst case scenario. And that was a short-term tox, biodistribution and tox study. Basically, in any of the animals, we didn't see any adverse effects that were directly related to the delivery of the vector. When we looked at the 1x, in the long-term animals, what we saw is that all of the vector genomes, or at least any of the tissues that registered a significant signal, they were all contained within the joint. There was no extra-articular genomes detected in those animals. So what's kind of interesting is that we've got multiple different bars here. Controls received no virus, and they didn't show any vector genomes in biodistribution. But the long-term animals and the acute animals would be these first 2 bars. And what fits with what we'd seen with the expression is we didn't see a drop-off from 2 weeks post-injection to a year. We didn't see any drop-off in the number of vector genomes that were detected in the articular tissues of the treated animals. And in these animals at the 1x dose, we didn't see any vectors, genomes outside the joint. However, in the 10x dose, we did have one horse that was an outlier. Of the three that were in that group, one of them, it showed as many vector genomes in the contralateral opposing joint as they did in the injected joint. We don't think it's an error of injection because there's an awful lot of virus to load into needles and deliver by accident. So it's sort of we've seen some sort of contralateral transfer of vector before, but we saw it here sort of in a large way. But other than that, that's basically all we saw with the farm tox study. So at this point, so just to summarize, we can deliver by AAV delivery. We can generate large amounts of IL-1Ra in the joint fluids. We can maintain them there indefinitely following a single injection. The transgene expression is greater in a disease context, which is a good thing. And the vector is largely contained at the site of injection. We don't see any adverse effects that are associated with treatment. On the upside, it reduces joint pain and improves some of the articular pathologies that are associated with the disease, which is the first time anybody's really been able to show that. And finally, so altogether, the data suggests that that AAV-IL-1Ra delivery is reasonably safe, and it's appropriate for testing in phase 1 trials. And we just, my colleague at Mayo Clinic, Chris Evans, just finally has jumped through all the hurdles and finally has final approval to inject the first patient. So we'll look to see how this study goes. It's a dose escalation safety study as well. So that brings me to the end of what I was going to talk about. I want to thank particularly Rachel Watson, who works in my lab. She's been the point person on virtually everything that I've shown you, as well as Pat Callahan and Andrew Smith. I'm not a veterinarian. These are my veterinarian colleagues, and particularly Andrew Smith. He was the guy that's done the heavy lifting when it comes to treating the animals. Thankful for the toxicology corps at UF, and then the various vector corps for helping us to generate enough virus to do the treatments. And then just appreciate the funding that we've received from NIAMS and NIH, as well as the Department of Defense and the CDMRP. So thank you very much for your attention, and if you have any questions.